Okay, so I'm going to start off with a slightly worrying statistic. 1 in 8 men in this room will be diagnosed with prostate cancer in their lifetime. It's quite worrying, but it's not exactly the problem. See, for most of you, you will be dealing with what we call pussycats. You'll have quite low-grade tumours that are slow-grown. They tend to respond well to therapy, so the survival on the whole is pretty good. For some of you, though, you're going to be dealing with tigers. These are aggressive, high-grade tumours that don't tend to respond well to therapy, and the survival for these patients is much more poor. And the truth is, we don't fully understand why that's the case yet. And that's really the essence of this project. We're looking at one of the factors that influences tumour aggressiveness. We want to see what it's doing in prostate cancer, why, and what we can do to stop it. So SIK2, or Salt Inducible Kinase 2, is an enzyme in your cells. Specifically, it activates other biological molecules that are important in cancer. And it does this through a process called phosphorylation. So we know that SIK2 is overexpressed. There's more of it in these aggressive tumours. We know that mutations in the SIK2 gene affect prostate cancer patient survival. If you look at patients who are suspected of prostate cancer, you can see that about 50% of them have some detectable levels of SIK2 in their blood. And those with the highest levels of SIK2 also have raised CD10, which is a proven marker for prostate cancer aggressiveness. In terms of the why, we know that SIK2 is regulated through pathways involved in metabolism, so energy production. Now, we already know that obesity, hypertension, cholesterol are factors that influence prostate cancer prognosis already. So part of this project is to pick out exactly which of these pathways SIK2 is involved in and which of these pathways regulate SIK2. We can do this through a variety of molecular biology techniques. Finally, a solution to this problem, I want to test I want to test inhibitors against SIK2, not just in cell models, but also in ex vivo tissue models, where we take a little piece of the patient's tumor, bring it into the lab and grow it where we can actually test the inhibitors on the tissue. Finally, we want to follow this up with some mouse model work to make sure that we can tell whether this treatment has efficacy and safety, and hopefully will provide a layer of knowledge that can go forward towards phase 1 clinical trials. And with that, I want to thank everyone who's helped me, and I should be below time, right? Wonderful, thank you.